Here we go. So you've been taking the Socratic quiz. Laboratory-produced GMOs are more dangerous than traditionally produced GMOs. True or false? There's no right answer. I'm just taking your opinion. I'm actually surprised it's this close. I'm surprised it's, what, 43% true, 53%. Despite what the quiz said, when I set up a true-false question in this program, it tells me I have to give a right answer. There is no right answer. But I'm surprised that it's this close. I'm impressed, actually, that there's this much variation still in the class. So that sets us off for what we're going to do today. About half of you think lab GMOs are more dangerous than traditional GMOs. About the other 50% of you must, I assume, think the opposite. I was really happy to find earlier this week, right, April 14th, just a couple of weeks ago, this happened. Somebody has now generated in the laboratory a mushroom that doesn't brown when you slice it. Finally! I've been waiting for this for years. Right? I don't want those stupid mushroom slices to be all gross and brown when I cook them. What did these scientists do? So they used a particular technique. I'm going to explain briefly the technique they used today. It's in the news all the time right now. It involves this enzyme called CRISPR. Well, it's the CRISPR-Cas9 system. Its full name, CRISPR-Cas9. It's a biological laboratory technique you can use to edit the genomes of organisms. How did they edit the mushroom genome? What does this say that they did? They deleted a handful of base pairs. In which gene? Polyphenol oxidase. Where have we heard about that before? That was last class. That was the Arctic apples, right? So the exact same target that a company in Washington targeted when they made apples that don't brown when you slice them. These people at Penn State just said, hey, we could do that with mushrooms too, I guess. So they used a specific technique to get rid of one of the polyphenol oxidase genes. So there are 6. They said they knocked out, that means got rid of, eliminated, removed one of the 6 genes that encodes this enzyme. And so now you get mushrooms that don't brown. So this is how this system works, very briefly. But you might be able to apply this to questions on an exam, for example. This cloud up here, the colored bubble, represents the enzyme. There's a specific protein, an enzyme, that's what's Cas9, is a protein. It's found in bacteria, and these bacteria use this enzyme whose job is to cut DNA. So that's the goal of this enzyme. Its only job, Cas9, is to cut pieces of double-stranded DNA, as shown here. So this protein in its natural state in a bacterium roams around the bacterial cell trying to identify foreign invader DNA and destroy it. It's like an antiviral response. This enzyme patrols the DNA that's inside a bacterium, and when it finds a particular piece of DNA, it cuts it in half. It cleaves both strands. It's an enzyme that destroys DNA. How would somebody who's a biotechnologist tell this enzyme where to cut? So you say you want to take out the polyphenol oxidase gene. This is the clever part, and this is why very likely in the near future someone's going to get a Nobel Prize for this technology, in all honesty. There's been a lot of discussion already about the individuals that basically commercialized this technique, taking this enzyme out of a bacterium and being able to apply it to engineer, modify the genomes of any species on the planet. It's actually pretty straightforward. Does anybody remember how a ribosome knows how to translate a codon, the messenger RNA, 3 nucleotides into an amino acid? What's the ribosome made up of? It's got proteins. like Cas9 is a protein, what other components are there of ribosomes? There are two different types of molecules that comprise the things that translate messenger RNA. They're part protein, and the essential part, without which they would not at all function, is RNA. So ribosomes have proteins, and they grab onto this RNA, and the RNA molecule reads the transcript and translates. That's where the translation happens, an RNA molecule that's part of the ribosome. And the same thing is true of this CRISPR-Cas9 system. There's what's here a gRNA, or a guide RNA. It's part of the structure of the protein. So Cas9 protein grabs this guide RNA, gRNA, and the sequence, if we blow this up here, this guide RNA will have a particular sequence. It's RNA, so it's A's, U's, G's, and T's. This is just sequence I'm making up, not necessarily the real sequence. But that sequence that's part of the guide RNA that's in the enzyme Cas9 will base pair, of course, with some sequence that's actually in the genome of the organism that this enzyme is exposed to. So UACAGC, for example, sorry, that should be a T, since that's DNA. A's base pair with T's. Okay. So TACAGC would be the DNA sequence that this enzyme RNA complex now recognizes. So this base pairing between the guide RNA and the double-stranded DNA is what targets that enzyme to a specific spot in the genome, and that's where it will cut the DNA. So what do you have to do if you want to remove polyphenol oxidase from an organism? How many times do you have to cut a piece of DNA to take out a gene? Say it louder. I heard the right answer. You all said the right answer twice. You have to cut once on either side of the gene, and then you can take the gene out. And that's what's shown in this middle panel. So you could have two different guide RNAs that have two different sequences that make this enzyme cut at two different spots on the chromosome. So what information do we need to be able to design a system to actually cut out a gene? You have to have two guide RNAs. What do we have to know to design those guide RNAs? Yeah, we have to know the sequence of the genome that we're trying to edit. So if I tell you the DNA sequence here, for example, just making up some random string of nucleotides, I could ask you, okay, what's the sequence of a guide RNA molecule that you would want to design and put into Cas9 enzymes to target it, to cut, to remove some stretch of this? Like, let's say that I was interested in removing those 5 nucleotides. And this is exactly what scientists are doing right now all over the world with this technology. You get a genome sequence, say you want to take out a gene. Say you want to put in a gene. Say you want to make a mutation at a specific spot. They're using this technology to go in and make very fine-scale changes, not waiting around for that crappy process of random mutation to occur over time. We can make specific mutations now in most organisms. All you have to be able to do is transfect the cells of that organism with a piece of DNA that encodes this protein, so that this protein, Cas9, will be expressed in the cell. And the enzyme is there. Give it the guide RNAs. That does the job for you. Take out genes, put in genes, induce mutations. So that's what these scientists did with this mushroom. Took out a gene in the laboratory using genetic modification techniques. So I want to give you a little bit of a handle now on what's going to happen in the rest of the class. We've got 2 examples of genetic modified organisms. Last time was more sort of big picture, GMO. I didn't make a very strong connection between that and genetics. Hopefully in this previous example I've started giving you that introduction into why genetically modified organisms actually has to do with genetics. So do these 2. Then hopefully with about 15 minutes or so left, I'm going to leave you with a survey that's an optional survey to take. I'm going to leave a little bit of time at the end of class for that process. So we're going to head into these last two examples of genetically modified organisms. Last class, I spoke about the evolution of maize. Mutation in teosinte that caused open fruit cups that allowed maize to evolve, or to be domesticated, rather, by farmers. The exact same story, or a very similar story, is true in rice. It has everything to do with genetics. What's an SNP? It's a type of mutation. Single nucleotide, yeah, exactly. Single nucleotide polymorphism. That just means one point mutation, a single base pair change. So what these scientists discovered was they were interested in the domestication of rice, how rice became a crop from its weedy wild relative. And so they sequenced the genomes of weedy rice, wild rice, and rice that's been harvested, cultivated. And they found that there was a single nucleotide change between weedy rice and cultivated rice that causes rice not to break, the actual seed itself, when you thresh it. When you try to get rid of the excess plant material and just get down to the part that we eat. Wild rice tends to break when you do that. You smash the rice on the ground or you thresh it. You'll break all the kernels of the rice, and then it's hard to recover from all the chaff. So there are two different types of rice that these individuals studied. One is a Japanese cultivar strain variety called Nippon Bear, right here. So it's non-shattering. It's one that's resistant. It's got high tensile strength, shown down here on the bottom. So what they're going to do is they're going to take a bunch of different strains of rice from different parts of the world, and they're just going to test the seeds in a physical test. What's the tensile strength of the seed? How much force do you have to put on it before it breaks? So they use a Japanese strain that's non-shattering, and then they use a different variety, cultivar, Casalath, which is an Indian strain variety that breaks. It's closely related to the wild ancestor of rice. And the point that they're making in this figure is there's this single base pair change. Non-shattering, you get a T, and in Casalath anyway, in this other strain, there's a G. The one base pair change. Is that convincing to you that one base pair change might cause this sort of a pattern? Take Indian rice, Japanese rice, sequence their genomes. I tell you, here's one base pair change. What should you say to me? What sort of questions would you want to ask? Why, pardon? Where did it occur? So like what part of the gene or where in the world geographically? Okay, so yeah, what is the gene? Does this cause an amino acid change, for example? What's the effect of the amino acid change? Very good. Are these the only two varieties of rice that have this mutation? Might be another question you'd want to ask me. Or then again, are there other differences in the genomes of these two strains? This can't be the only one. Rice genome is huge, millions of base pairs long. And this is not the only difference between these two varieties of rice, the Nippon Bear and the Casalath. So what do? What would you do to answer those sorts of questions? So you've compared two strains, two varieties so far. What would you do next? How many more rice varieties are there? Let's look at some more. See if this pattern is always the same. So could you take any non-shattering rice? Does it always have that same non-shattering nucleotide, the T? And if you take any wild rice and you look at its genome at the exact same spot, does it always have a G? That would give you a little bit more information about whether or not this pattern is consistent among all rice varieties. And so this mutation, this one position here, is what they've called QSH1. So they looked at that specific spot on a chromosome in rice in 8 different strains of rice, shown here in this table down below. There's B's and A's now instead of C or T's and G's. But look across here now. Here's all the ones that have a B. That's one of the two nucleotides. You'll have to tell me which one B is. What's the tensile strength on their seeds that are necessary to break it? How much force is necessary? Little or a lot? So non-shattering degree, you've got not very much force necessary. You've got a lot of force necessary. So this is like a shattering strain. These tall bars represent non-shattering. So is there a pattern here between genotype in this column of B's and A's and phenotype? When you look, all 5 of these that have a B, that happens to be equivalent to a G nucleotide, are they shattering or non-shattering? When you look at tensile strength, it doesn't take a lot of force to break the seeds. They're a shattering type. Right? All 5 of those strains that have a G from different parts of the world are all shattering type rice. What happens when they have the A, which is equivalent of up here, the T from the non-shattering cultivar? 3 different varieties of rice. They're all non-shattering. It takes much more force to break their seeds. So this single mutation is responsible for this one phenotypic change in basically all varieties of rice in the world. Just single base pair change. We could just as easily do this in the laboratory, but this wasn't done in the laboratory. How did this genetic change occur? T versus G. What happened? How did we get non-shattering rice? What happened? Selective breeding? Absolutely. So there could be multiple different traits that somebody's looking for. Number of seeds, right? So you want a rice plant that grows lots of grains. So what was happening was we were just waiting for mutation to occur. And when the right mutation occurred, somebody in the rice paddy was there at the right time in the right place to find that rice plant that had this random mutation to start selectively breeding it. So what's the benefit of genetic modification in the laboratory? What's one benefit of in the laboratory genetic modification?
You don't have to wait.
It's faster, presumably. So one of the potential benefits of doing laboratory-based genetic modification is, yeah, you don't have to look at 10 million rice plants to find the one that has the random mutation that causes the phenotype you're looking for. And I will not walk you through this slide as well. It's just more data that shows the same pattern. You look at different rice plants from different parts of the world, Chinese rice, Japanese rice, Indian rice, they all share the same pattern. It's almost like there was one ancestor of all domesticated rice, which is probably actually true. So that's a potential reason to use laboratory-based genetic modification techniques. You don't have to wait for random mutations to cause the effect that you're looking for. You just try to make it yourself. The point being that rice, this one trait, just required one single mutation. So I'd like you to reflect on, please, I'm only asking you this because I'm interested in developing students who are excellent ambassadors of information out into society. Would it be any different if that mutation had happened in nature or if I had made that mutation in a laboratory? Single base pair change. So you don't have to answer, but maybe keep that in the back of your mind. So is GMO in the laboratory equal to GMO in the field or not? There's sometimes that it basically is the same thing and sometimes it's not. And that's where what we're gonna talk about now is when maybe not to go genetic modification. Unless there are any questions at this point. The rice example is just to show an example like corn of how easy it is to get a phenotypic change, single base pair change. So then we get to the when GMOs go bad. Has anybody here heard of glyphosate? A couple of you, a few of you. It's an herbicide. It's also known as Roundup. This is the target of crops that groups like Monsanto, big GMO organizations, evil empire sort of thing, make. So I'm not saying that they're bad, but this company is well known for producing crops that are called roundup-ready crops. All of their crops that are Roundup ready have, as it says over here, they express a little bit more of a protein that the plant does. The plant has a gene in it that degrades this chemical that's an herbicide. Normal mode of action of glyphosate, kill plants. It disrupts photosynthesis, if I remember correctly. So if you're a plant and you've got glyphosate, you die unless you happen to be engineered with resistance to this chemical. You put a gene in, the gene produces a protein, the protein degrades the glyphosate before it can interfere with photosynthesis. So if you're a farmer, the idea is you get this GMO seed, genetically modified organism seed from Monsanto, you plant it in your field, and then to kill all the weeds, what do? You spray Roundup. And what happens to all the plants? What happens to the weeds? They're not GMO. They're just plants sitting out in your field. They don't have this transgene, so the Roundup kills them. What does the Roundup do to the crops that are GMO? Nothing. That's the idea. That your plants that you're trying to grow are resistant to the thing you're using to kill everything else. What's one of the big concerns that a lot of people have about this sort of genetically modified organism? Carries a gene that may not be, I'm not saying in this case that it's true, but it may not be from the same species. And it's a gene that confers an advantage on the plant that has it. In this case, you make a transgenic plant, put it in your field, it resists death by herbicide. What's a potential issue? Bingo. So what happens if Roundup Ready rice, for example, hybridizes, makes offspring with wild rice, rice that's growing out in nature? So then now all of a sudden you've got wild rice that is resistant to this chemical. What happens if that wild rice crossbreeds with its closest relative and so forth and so forth and so forth, and this transgene, this genetically modified organism, this Roundup resistance gene spreads? What happens if it spreads into weeds? Just by hybridization, by plants having sex with other plants and passing genes around. That's one of the big fears of a lot of people that don't think that this sort of genetic modification is appropriate, at least to put out into the field where pollen can blow all over the place, and you can't really control who is having sex with who in plants. It's pretty hard to control plant sex. Have you tried it? Anyway. So these are some common issues. And the responses tend to be this. So the pro-GMO argument for this sort of like Roundup Ready crops is what you can read up here, but don't read the text, it's boring. If we put a gene that produces a protein that detoxifies glyphosate into a plant, and there's no herbicide present, what do you think the plant is going to, is the plant going to be healthier or less healthy? So it's herbicide resistant, but there's no herbicide around. Is it going to have the same health? Will its health be better or will its health be worse? So don't just tell me what I want to hear. What do you think? So okay, we have some people think that it's not going to matter whether the plant's transgenic or not. If there's no herbicide, it doesn't matter. Other?
No, I agree with that.
You just agree. Okay, so it doesn't affect the fitness. Others?
It will affect the fitness.
So it would be healthier or less healthy?
Less healthy.
And why would you argue that?
Because there's, from what I know about plant science, there's always
Okay, so you're making, the argument goes along the same lines that if you're a plant and you're putting energy, we've heard this story before. If you're a plant and you're making, same thing as lactose, the galloperon. Why would a bacterium want to make all these enzymes that process sugar if the sugar's not around? Is it cost to the bacteria to make these proteins when they're not necessary? So the argument from the pro-GMO camp is that, well, of course, this gene will never spread into the wild relative. It could, maybe, by hybridization, but if the gene gets out, it's not going to go anywhere because it's not advantageous to plants that have this gene if you're not actually spraying herbicide on them. Because if you're a plant and you're making this transgene and there's no benefit to you of making it, your cells you might be slightly less healthy than other plants because you're spending part of your energy, instead of growing or photosynthesizing or making seeds, you're spending part of your energy making this enzyme that detoxifies a chemical that's not around. And that's an argument, again, that the pro-GMO camps have made.
So couldn't it also still just be carried around as a recessive gene?
Right, so it could, right, so this gene could still spread. That's an excellent point. So the gene could still spread from plant to plant under the assumption that it's neutral. That is, it doesn't help or hinder the plant. So if this addition of this gene does nothing to the health of the plant, yeah, you'd expect it to spread and you'd never know, unless you started spraying Roundup on everything. But in the case back here where there's a detriment, where it's actually not beneficial to have this gene, then you'd expect it not to survive in the wild. The only plants that like having this transgene are the ones that are actually having Roundup sprayed on them. So that was sort of the state of the field, no pun intended, for quite a while, and then this study came out. So the assumption, again, to summarize, was that there's no benefit to a plant just being transgenic. That is, being transgenic in itself, having had a gene added, is not beneficial in general. It's only beneficial when you have Roundup around. Until this study. And this is just one study, so keep that in mind. This is one study that scared the pants off of people. Not literally, figuratively. Scared the pants, never mind. So each of these black bars represents a transgenic strain, so a Roundup-ready crop. And each white bar is the same strain minus that gene, minus the transgene. Transgene, I'm sorry, I didn't define, is the gene that's been put, i.e. trans, the gene that's been put into an organism, transgene. So what are we measuring here? We've got four different pairs. We've got a transgenic and a non-transgenic, otherwise genetically identical, this pair. Same here, transgenic, not transgenic, otherwise genetically identical. This one just has the gene, WH3, WH4. So we've got 4 independent experiments. What are we measuring? Y-axis. What are we measuring? Okay, is that, what's that a measure of? Not chemically, but why do we care about photosynthesis in these plants? What are we trying to ask? What's the question?
Primary production?
Well, primary production, so yeah, is the plant healthy? Is it producing or is it not? Is it happy, is it unhappy? Which plant's happier? The black bars or the white bars? Is photosynthesis good or bad? Okay, good. So all of these plants, no, don't make me, oh, you made me do it. Okay, so the black bars are all happy bars, right? Compared to in each of these 4 independent trials, the plants that were transgenic photosynthesized more than their genetically identical non-transgenic siblings. That's not good. It means the only difference is these plants have the transgene, those plants don't. This is in the absence of the herbicide, by the way. Transgenic plant, non-transgenic plant in a field, no herbicide. So you're not even trying to kill this plant. They're sitting in the same field, same conditions. photosynthesis is higher in the transgenic plants. What are we measuring here? Number of seeds. So what happens? If you're the transgenic plant, are you better or worse off than your non-transgenic sibling? Better. You make more seeds. You're more fit. Holy shit. The whole idea was the transgenic plant's not going to care if it's transgenic or not unless you're applying herbicide. No herbicide? By itself, it seems in this case, in this one study, just one study, in this one system, that if you're transgenic, by itself, being transgenic makes you healthier. Having a scientist having tinkered with your genome somehow made them healthier. Same was true if they looked at, not only did you make more seeds, but a higher percentage of your seeds germinate and make more plants if you're transgenic, even without the herbicide. So then everybody sat back and took stock and said, you know, maybe we should be a little bit more methodical about how we test genetically modified organisms before we think about whether or not we're going to actually put them out in nature and grow them as crops. Now I want to make it clear, just because I've seen this in some of the comments, and thank you very much for all the comments you left at the end of last class about why you feel the way you feel about your opinion on genetic modification. One of the concerns occasionally is that are there plants that have actual herbicides or pesticides built into the crop, and do we want to eat those? This is not an example of that. This is an example of a naturally occurring protein that degrades the herbicide being put into a crop. This is not a pesticide being produced by the crop itself. I want to close with the counter-argument, after I clean up a little bit. I'm going to close with a counter-argument. So half of you said you thought the GMOs were no worse than traditionally bred, no less safe than traditionally bred crops. So I want to leave you with an argument that makes that point, that sometimes even traditional breeding is dangerous. Traditional breeding could kill you. This is how. This is all from a study that I just showed you the reference for. Skip past it. Does anybody recognize these fruits? What are these? So we've got bell peppers up here in the upper left. What are these white things? Eggplants. We've got tomatoes. What are these funny tomatillos? Yep, related to gooseberries. Bless you. How are all of these related to each other? They're all from the same plant family. They're all from the pepper family, which is also the potato, tomato, gooseberry, tomatillo, bell peppers, eggplants. It's a plant family called the Solanaceae. I wish Dr. Constable was still here from the previous class. All of these plants produce this poison at some level. At some point in their life, in some part of the plant, they produce solanine, which is deadly to us. You eat a little bit, you get diarrhea. Eat a lot, you die. Okay. Tomatillos, tomatoes, peppers, potatoes. And you may not know a lot about potatoes. I'm not pretending that I do know a lot about potatoes, but I do know what I've read, which is that different potatoes have been bred for different qualities. Of course, there's the potatoes of different sizes. You've got the baker potatoes, and then you've got the little cute little red tomatoes that you boil and put a little butter and parsley on. You've got the fingerling potatoes that you, whatever. I like to cook. Some potatoes are bred for being chipping potatoes. So you've got good starch content. When you slice them, they don't break. When you fry them, they achieve a perfect color, according to the food industry. So the food industry actually has a color scale. It's a rating scale, 0 through 10, and you hold it up next to the chips when you fry them, and then you train people to say, is this potato good or not for making the perfect color potato chip. So for decades, centuries, people have been breeding potatoes to do different things. So in the ever demanding and ever increasing urge for people to buy potato chips, I guess, the breeding for chip potatoes has been going on for decades. A few decades ago, somebody came up with the perfect, ideal potato to make chips out of. Crisps, if you're British. Anybody British? Anyway. Chips or crisps. Right starch content, right type of starch, right color when they were fried. That was called the Lenape potato. That was its variety name. That's represented here in two different spots on this table. There are two different Lenape varieties right here, circled in blue. And they started marketing this potato, and they grew a lot of the potato, and they started shipping it out to be turned into potato chips. And somebody then so this Lenape potato was just traditionally bred. They were hybrids of different potato varieties that people had already been growing. Then somebody thought, you know what, we should probably feed some of this to a rat. Just to make sure it's safe. Rat died. All the rats died. So then in this paper in 1980, this happened well before 1980, but this paper from 1980 shows what happened. What happened? How does the Lenape potato differ from these other potato varieties? When you look at the amount of solanine in milligrams per 100 grams of protein produced, what's happened to the Lenape protein, the Lenape potato? They make like 10 times more solanine than any other potato plant. They make levels of solanine that will kill you if you eat these potato chips. And they didn't happen to check apparently this one person thought, you know, maybe we should feed some rats. Just a good thing. So this potato actually never went to market. How did this potato get produced? Is this GMO? What do you think? Depends on what definition you came up with last class. Is this GMO? Is the Lenape a genetically modified organism? Yes, but naturally. It was created through traditional crossbreeding programs, taking different varieties of potatoes and making new hybrids, combining different traits. What happened in this case? How did the Lenape get so poisonous? All of a sudden you get this monster potato that produces like 10 times more solanine than other potatoes. It's because the parent strains, the parent varieties that were used to produce this hybrid, the Lenape, had particular combinations of mutations that when they were brought together into the same organism, ramped up solanine production. Chance, basically. The wrong combination of mutations at the wrong time. The potato plant didn't care. We would have cared. And so the take-home message being that it's not necessarily the case that when we make GMOs, we don't know what's going to happen. That's usually true. So we don't know if we make a glyphosate-resistant plant and put it in the wild. We don't know if its transgene is going to escape into wild populations. But it's equally likely that by doing conventional breeding, you make some random combination of mutations that does something detrimental. So we have to be careful, regardless of which type of genetic modification we're doing. Traditional breeding or specific mutations. Both can have unwanted side effects. I'm going to leave you with a couple of closing remarks to push us into what we're going to talk about next class and beyond. But before I do that, are there any other questions or comments that anybody wants to make about genetically modified organisms? Please.
So, naturally and man-made, but for man-made, then at least you can control what gene it's doing, but for natural, then so why is gene level bad?
So that's exactly the question. So the comment up here was, with conventional breeding or within the lab, maybe one perspective is, it's better to do it in the lab because we know exactly what genetic change we're making. Whereas in the potato example, the idea was that you don't know what combination of genes. When you're dealing with combining whole genomes at once, it's harder to predict what the outcome is. If we make a single specific mutation at a time, maybe we can better control or better study the potential outcomes of just that one mutation. So then the ultimate question that was asked was, why would labeling GMOs be important? And that's exactly why I'm raising this. And I don't assume that we're going to come to a conclusion among the class, but it might be something that's worth thinking about on your own. Should we have to label GMOs? Is there anything, in fact, I can challenge you, we do this in some semesters and not others. We didn't do it this semester. Next time when you go home, look in the refrigerator, look in the cupboard, if you go to the supermarket, find a food that's probably not genetically modified. In either way, either conventionally bred or transgenic. Try to find a food that's never been tinkered with, that's being sold for our consumption. Okay. So next class we're going to start talking about epigenetics, which is something, it's a type of genetics that has nothing to do with DNA, almost. This is an example of epigenetic regulation of genes. So everybody, calico cat, it's got some patches of its body that are one color, some patches of its body have a different fur color. So that difference in phenotype is not due to genetic change. That is, all of the cells in this cat, it's a single organism, all the cells have the same DNA sequence. So what causes the difference in color? All of those cells have the exact same DNA. Some of its cells make black pigment, some of their cells make a different color pigment. What's going on? So it has something to do with X chromosomes and bar bodies. But the X chromosome still has the same DNA sequence. So we're going to be talking about what happens not to the sequence of DNA, that's genetics, what happens on top of the DNA sequence, or epigenetics. This is basically another version of the same point. How many of you know a set of identical twins, personally? If you've met some before, you've seen some in the movies. Harry Potter fans, anybody? Okay. So I know several sets of identical twins personally, and I know I can tell them apart. Right? There's subtle differences. Their voices are a little bit different. They appear different. They're identical, but they're not quite identical, in my opinion, in my expertise. And the same is true Fred and George. Same situation as the cat. Genetically identical at the DNA sequence level, yet different. So what's the basis for those differences? That's what we'll talk about next class. So, in the last 5 minutes, please go to the Blackboard website. Either use the Blackboard app or more likely use a web browser. Go to the Blackboard website. And there's a link that I added in the course documents folder, in the URLs folder,